Good evening, everyone. This is your first week session with me. So just let me know, are my slides visible and am I audible? So I'll start with the session. Will you please drop me a text on the chat box that am I audible? Okay, thank you so much. So I'll start with today's session. So I am Dipanshi and I am a PMRF scholar. So I'll be taking your doubts, and in this session we'll be going through the week 1 topics, in which we'll see that what you have covered in week 1. Along with that, we will practice few MCQs, and we'll also look into the current literature, that what are the studies which are currently going on, and then I'll take your doubts if you have regarding the assignment or any other doubts. So now let's start. So first of all, this session is about the genetics and genetic engineering. So before beginning, we first must understand what the gene is. So gene is a functional unit of heredity, and it is a DNA sequence which encodes either a protein or a RNA molecule. So not all genes give rise to protein sequences. There are some genes which also give rise to the RNA molecules which helps to form the proteins, such as rRNAs and tRNAs. So these genes, they are inherited from both the parents, both mother and father, and they determine the traits. So traits means characteristics, that what a offspring will have a characteristic. So that is referred to as traits, such as eye color, blood type, or disease risk. So certain disease are also inherited from genes. And then comes genome. So gene is a single term. When we talk about genome, it means we are talking about entire set of genetic elements, or all the genes which are present inside an organism. So if we talk about eukaryotes, then genome is mostly stored in the nucleus, because we have well-defined nucleus. But when we talk about the prokaryotes which do not have nucleus, so in that case they have the genes or the genome which is present in the cytoplasm. So that is one of the difference among eukaryotes and prokaryotes. And along with this, if we talk about eukaryotes, then they don't have gene only present in nucleus. We also have gene in our mitochondria, so that is known as mitochondrial genome. And if we talk about plants, then they have genome in their chloroplast in addition to the nucleus. And the term genome was given by Hans Winkler in 1920. So here I have shown that how when we zoom into the cell, it has a central body which is nucleus, which contains chromosomes. And these chromosomes, these are the DNA molecules which are wrapped around the histone proteins into compact structures. So that are chromosomes. When we further elaborate this chromosome, then it is composed of DNA. And these DNA, they have small gene fragments. So in this way, genes are spread across the entire DNA sequence, which combines with histone to form a chromosome, and then multiple chromosomes are present inside the nucleus of a cell. So this is how the genes are arranged inside the cells. Then there are two terms which are most frequently talked in genetics, which are genotype and phenotype. So genotype means what is the genetic makeup of an organism. When we are talking about the genes which are present in an individual, so that is referred to as genotype. And when we say phenotype, so it refers to the observable characteristics, that what are the results of those genes, how they are expressed, what we can see. So this phenotype means which we can observe. And these terms, genotype and phenotype, they have been termed by Wilhelm Johansen. So he was the one who introduced these terms. Then there are some historical milestones which we will see that how this field has progressed through years. So first of all, as we know, the father of genetics is our George Johann Mendel. So in 1865, he has described that there are certain factors which could be heralded through a pea plant experiments. So at that time, he did not know the term gene, so he called them as factors. And then in 1909, when other experimentalists came up, then Johansen was one of the scientists who mentioned that these factors are the genes. So he gave the term gene to these factors. And then in 1920, there was another scientist, which was Hans Winkler. So he introduced the term genome. He said that these genes, they are present in the set. They are combined together and there are multiple genes which are present. So they are referred to as a genome. Then in 1928, the Griffith was a scientist. So people were trying to find out that what is the thing which is leading to the inheritance. So is this a DNA or is it a protein? So what is leading to this inheritance? So 1928, when Griffith came, so he mentioned that there is a transforming principle in bacteria. So he was studying different strains and he was checking that what is responsible for this inheritance. So he called that there is a transforming principle which is transforming from one bacteria to another bacteria and which is leading to change in the strains. And then in 1944, other scientists came up such as Avery, MacLeod and McCarthy, who have confirmed that DNA is the genetic material. So they further have performed different experiments with which now it was confirmed that DNA is the genetic material. And later on in 1952, Hershey and Chase, so they were the final set of scientists who validated that DNA is the molecule of heredity. So these three group of scientists, they worked on making the hypothesis, or who has validated this hypothesis that DNA is the genetic material which is responsible for the hereditary traits. And then in 1953, the studies began onto this DNA, because as we know that the traits could also be the disease traits, or if we can control the traits, then we can do multiple things. So to understand it in detail, Watson and Crick came up and they tried to look into the structure of DNA, how it looks and what it is composed of. So there were other scientists as well who were working on the DNA sequence by looking at the X-ray crystallographic structures. But then Watson and Crick were the first one who proposed the double helix structure of DNA molecule. Whereas there was another scientist, which was a lady, Rosalind Franklin. She was the one who was having the X-ray image of DNA molecule, and she was not able to completely interpret that it is a double helical structure, but Watson and Crick were able to interpret it. So they were the ones who were given the title that they have published the DNA double helical structure. Then in 1972, Paul Berg, he created a first recombinant DNA. So after DNA has been discovered, so people tried to look into the detail that how can we control the DNA, what are the additions which we can do, how we can enhance the traits. So then came the concept of recombination, that we can recombine this DNA with the gene of interest and we can increase its copy, and then we can change the trait or we can design certain drugs. So Paul Berg was the first pioneer who was the first person to create the recombinant DNA molecule. Then 1973, Boyer and Cohen, they developed first transgenic organism. And then in 1978, recombinant human insulin was also produced by the company Genentech. So in this way, this is how the historical milestones were present, so with which now this field has advanced a lot. And we can see that in 2003, human genome was also completed. So now we have the information of entire human genome, that what are the different genes, and now we can study that which genes are responsible for disease and how we can treat them. So this all has helped us into the field of personalized medicine, gene therapies. So now in this today's session, we will be looking into the basic concepts, that how it all started. So before moving further, we will look into the structure and nature of DNA molecule, because it is the basic component which we are altering. So first of all, there was a scientist named Chargaff, and there are certain rules which he has given. So according to Chargaff's rule, he mentioned that number of purines are always equal to number of pyrimidines. That means we have two purines, adenine and guanine. So he said the amount of adenine must be equals to the amount of thymine, because when we look at the DNA structure, then adenine base pairs with thymine. So if they are not in equal proportions, then it will lead to the breakage of double helical strand of the DNA. So in the next slide, we'll see the double helical structure of DNA. But for now, just keep this thing in mind, that amount of adenine should always be equal to amount of thymine, and similar goes to the guanine as well as cytosine. So in this way, if we further sum the number of adenines and guanines, then their sum must be equals to the sum of thymine and cytosine. So this is the rule 1 of the Chargaff's rule. Now the second rule is that amount of adenine plus thymine. must not be equals to amount of guanine plus cytosine. So there is little variation among these proportions, and this proportion varies among different organisms. For example, if we talk about the prokaryotes and different eukaryotes, then they have different proportions. Some organisms which are found in the extreme conditions or thermophilic conditions, they have higher amount of G plus C content. So I'll just give you a basic idea that G plus C, so guanine and cytosine, they have triple bond between them, due to which this bond is very strong, which cannot be easily broken up. So due to which the primitive organisms which were extremophiles, which were present in extreme conditions, so they generally have greater number of GC in their DNA, due to which their DNA was not disrupting so fast in the extreme environment. So this is one of the reason that if we explore this content, then we can figure out that what is the nature of organism, where it could be present. But it could be same in different tissues of the same organism. So these are two rules given by Chargaff. Now we will look into the structure and nature of DNA. So the model which is given by Watson and Crick, in which we will understand that what are the components of the DNA and how it is arranged. But before moving further, I'll just check. Okay, so if you have any doubts, then let me know in the chat box. I'll check and then I'll take your doubts as well. Okay, so now let's begin with this Watson and Crick model. So James Watson and Francis Crick, they were the two scientists who have given this model, and they have also received Nobel Prize in 1953 for this model. So they said that this DNA, it is a double helical model. That means it has two strands which are running anti-parallel to each other. So this DNA, it has the directionality. It runs from 5' to 3' direction. So towards its 5' end, it has a phosphate group, and towards its 3' end, it has a sugar. So it has deoxyribose sugar, in which the third position carries the hydroxyl group. So this OH group determines the 3' end of DNA. So now it has two anti-parallel strands. So one is running from 5' towards the 3' end, whereas the other strand is running from 3' towards 5'. So they are anti-parallel, and then each strand serves as a template during the replication. So when replication happens, then the double-stranded DNA, it denatures. It forms two single strands, and those two single strands, they serve as a template on which the complementary RNA or complementary DNA molecule will be synthesized. So that is referred to as template. And their model was based on critical X-ray diffraction data, which was generated by Rosalind Franklin. So she was the lady who has shown this data to these two scientists, and they were able to interpret that this X-ray pattern is like a double helical pattern. So they were the one, but their analysis is based on the X-ray diffraction data. So now let's see what are the different properties of this double helical DNA molecule. So it has in its backbone, it has sugar phosphate present. So sugars are linked together by the phosphodiester bond, and there is a phosphate group which is responsible for this phosphodiester bond. So if you can see, this is my third carbon of my deoxyribose sugar, and deoxyribose sugar is present in DNA, whereas in RNA we have ribose sugar. So the one difference is that in its second carbon we don't have OH group, we have simply H, due to which this position is deoxy. That means it is lacking oxygen. So this is the difference between DNA and RNA, one difference. So now in this 3' end or third sugar, it has the OH group which makes bond with this phosphate group, and it combines to the fifth carbon of the ribose sugar, which is the next sugar. So this is my first sugar, this is my second sugar, and it is joined by a phosphate group, and this type of bond which links two sugar moieties together is known as a phosphodiester bond. So in my double helical strand we have phosphodiester bond in the backbone where the sugars and phosphates are present. Now each sugar is linked to a nitrogenous base, which could either be purine or pyrimidine. So in this case we have adenine, which is a purine. So now adenine can only pair with thymine. So in its another strand which is complementary to this particular strand will have thymine in it. Then only there will be a base pair, means this bonding, then only this hydrogen bonding is possible if they have the complementary nitrogenous bases. So adenine is forming double bond with the thymine, and this bond is present between the hydrogen and oxygen. So this is a hydrogen bond, and hydrogen bond is present between the hydrogen and the electronegative element. So this is one of the feature of hydrogen bonds. And now if we talk about the another nitrogenous base, then it is cytosine, which base pair with guanine, and in this case there are 3 hydrogen bonds which are observed. So if we compare among adenine, thymine as well as cytosine, guanine, then this CG has triple bond, due to which these bonds are difficult to break. So one more fact which I will tell you is that whenever there is a replication taking place in which we need to denature the double-stranded DNA and we need to convert it into a single strand, then you will see that the origin from where the replication starts, it will be having greater number of AT residues. It will have greater number of the nitrogenous bases which are composed of adenine and thymine, because these double bonds are easy to break. So denaturation is easily possible at the sequences where AT is rich. Whereas if there is CG, then it's quite hard to denature that particular DNA segment. So yeah, and then if we talk about the diameter of this double stranded DNA, it is 2 nanometers. Then there is a major groove as well as minor groove. So the groove which is deep as well as wide, it is known as major groove, whereas this particular part which is narrow and shallow, it is the minor groove. And if we talk about the entire turn, that how many nitrogenous bases are present in one turn, so there are 10 nitrogenous bases, so you will observe 10 such bonds, and this particular length of one such turn is 3.4 nanometers. Okay, so I hope you are aware of this nanometer, angstrom. So don't get confused. In many books, this double helical DNA is labeled with respect to angstroms, whereas in other books it is mentioned with respect to nanometers. So when we talk about angstroms, it is 10 raised to power minus 10, whereas if we talk about the nanometers, it is 10 raised to power minus 9 meters. Okay, so this is all about the DNA structure and its nature and what are the constituents. And one more thing is that the DNA has adenine, thymine and cytosine, guanine. Whereas if we talk about RNA, then in place of thymine we have uracil. That is one other difference between DNA and RNA molecules. Now as we know that DNA is the primary component which is responsible for the genetic traits, but now what this DNA is doing? So this DNA is getting converted to RNA and it is carrying a signal, due to which it can form proteins. So now from how DNA is deciding what protein will be formed, so this is present inside a genetic code. So genetic code, it is a set of rules which governs that what will be the protein that will be synthesized, and it is based on the nucleotide sequence. So now my DNA will convert into mRNA, which is the messenger RNA, and mRNA is the one which further forms proteins. So DNA can also give rise to tRNA as well as rRNA and several other types of RNAs which are responsible for other constituent synthesis. So now mRNA is called messenger RNA because it is carrying the message that what protein need to be synthesized. So now these mRNAs translate into amino acid sequences. So as if we talk about DNA, it is made up of sugar, phosphate, nitrogenous bases. Similarly, protein is composed of amino acids. So there are codons which are triplets of nucleotides. So when we count 3 nucleotides, it is referred to as a codon. Each specify that what will be the amino acid that could be synthesized during the translation. So translation is a process in which mRNA is converting into a protein. And now these codons are specifying what amino acid could be synthesized. So now there are total 64 possible codons, out of which 61 are synthesizing amino acids, whereas 3 codes for the stop signals such as UAA, UAG and UGA. So these are the stop codons. And we also have a start codon which is AUG, which synthesizes methionine. So that is the start codon from where the translation will begin. Now this code is nearly universal, with which we mean that it will be universal, that whenever that particular codon is coming, it will always give rise to the same amino acid in all the organisms. So that is referred to as its universal nature. But different experiments have mentioned that in different organisms, such as if we compare human and yeast, there are certain codons which can be different. So there is some codon changes, ambiguities which might be present if we compare different organisms or different organisms. And then it was deciphered through a groundbreaking experiments by Marshall Nirenberg, Hargobind Khurana and Philip Leader. So these are three scientists who have worked to identify this genetic code and who have also received Nobel Prize. So Nirenberg, Hargobind Khurana, they have received Nobel Prize in physiology for the discovery of this genetic code. And this is universal, it is non-overlapping and degenerate. So there is this one thing that these three nucleotides, they can make one single amino acid, but if we talk about that particular amino acid, so it can be coded by multiple codons. One codon can only synthesize one amino acid, but one amino acid can be coded by multiple codons. So this thing is known as degenerate, which allows multiple codons to code for the same amino acid. So as you can see in this case, for example, let's take example of serine. So serine can be coded by UCU, UCC, UCA and UCG. So these all codons can synthesize one single amino acid, that is serine. So this nature is known as degenerate. Now in humans, the translations always begins at the start codon, which is AUG, methionine, and it ends at the one of the three stop codons, which are UAA, UAG and UGA. Now one more thing which I want to tell you with this is that the start codon, which is the, you can also call it as a canonical start codon. That means by default the translation always begin with AUG. But if we talk about the current system of research, they have identified that other than this canonical start codon, the translation can also begin with non-canonical start codons such as GUG or CUG. So they are alternate start codons. So in many cases the scientists have discovered that it's not the fact that it always begin with AUG. In some scenarios, GUG can also act as a start codon. And the stop codons are these, UA, UAG and UGA. These are fixed. Whenever they will come, the translation will stop. Now we will move to genetic engineering. So is everything clear till now? Okay, now let's start with genetic engineering. So genetic engineering is the lab-based modification of DNA. Okay, thank you. Yeah, so it is a lab-based modification of DNA to alter or enhance the traits in organisms, and it can also involve gene additions, deletions or editing. So when we say term engineering, that means we are doing something, we are erasing, adding, or we are doing some kind of technical things with the genes. So that is genetic engineering. And the applications is that we can produce different biopharmaceuticals such as insulins, vaccines. We can design certain GM crops, which are genetically modified crops, and livestock. So when we say livestock, that means we can add certain genes, we can engineer some genes in an organism such as sheep, goat or some cattle, such that the milk which they give, they might involve those proteins, so which can help us further to synthesize that particular respective protein with the help of that particular livestock. Then comes industrial enzymes and fermentation. So this industrial enzymes can also be synthesized with the help of genetic engineering, because we know that these bacteria, they replicate quickly, they have very short lifespan, so they are replicating quickly, there is fast cell division, due to which they are good targets to synthesize proteins quickly. So that's why they are used. If we look at the overview of genetic engineering, which is also referred to as recombinant DNA technology, in which we are recombining the DNA. So we generate the DNA fragments. First of all, we must have idea that which protein we want to synthesize. Then we must know which gene is responsible for that protein. So we select that particular DNA fragment which give rise to that protein. So that particular DNA fragment is obtained, and now that DNA fragment is broken down with the help of restriction enzymes so that we can add it into the plasmid. So plasmid act as a vector which can incorporate this DNA of interest into itself, and then we can insert this particular recombined DNA, which means the plasmid and DNA of our interest, into the prokaryotic organism or the host from which that plasmid was isolated. So this is whole procedure of recombinant DNA technology. In the later slides, we'll look into the detail, each and every step, how it is done, but this is the overview. Now what are the steps? So these steps we have already discussed, that we identify gene of interest, we isolate that, we clone it into a vector, that means we put it into a vector. So this is the vector which is acting as a vehicle which could take up the gene of interest. So this is my vector. Now we transfer it into the host organism, because this recombinant DNA is synthesized, but we also want multiple copies of this DNA. So we need to put it into the host, which will act as a machine which will synthesize more copies of this recombinant DNA. So this is our host, and then we confirm the expression of the gene product. So after this division has been done, multiple copies have been produced, then there are certain experiments which have been done to make sure, to check that whether the synthesis has been done or not, and how many such recombinant plasmids which are present inside that organisms. So those are confirmed after this replication, rounds of replications being done. Now there are different molecular tools which are used in this genetic engineering, because these are the straightforward steps. But now what are the different tools which are used to follow these steps? So one such tool is restriction enzymes. So these restriction enzymes, we can call it as a base of genetic engineering, because if they are not present, we cannot do genetic engineering. So these were discovered in bacteria as defense mechanisms against phages. So phages, or you can also call them as bacteriophages. So these are the viruses which attack the bacteria. So now these restriction enzymes were discovered by accident. So there was a scientist named Kurenberg who was studying, and he realized that bacteria were fighting with the viruses. They have some mechanism due to which they were able to recognize that what is the foreign DNA and what is their own DNA. So then this restriction enzymes were identified. Then they were categorized into 3 types. So one is type 1, which cut randomly anywhere and they are multifunctional. Then comes type 2 restriction enzymes, which are most widely used and they cut at specific recognition sites. And then type 3, which cleave at a distance from the recognition site. So now before talking about them, I'll also tell you about what the restriction enzyme means. So there are 2 types of enzymes in our body. One are the endonucleases, other are the exonucleases. So as the name suggests, nuclease, that means the enzyme which can break down the nucleic acids. So they are nuclease. Then endo and exo. So endonucleases means the enzymes which can break the nucleic acids from within, from inside. Exonucleases means the enzymes which can break these nucleotides from the terminals, from external positions. They cannot make the cut in between. So those are exonucleases. So when we talk about restriction enzymes, they are the endonucleases. That means they can cleave the DNA from within. So these restriction enzymes, so how they are different from the basic endonucleases, why they are called restriction endonucleases or restriction enzymes, because they have a sequence, a recognition sequence, which they recognize and then they decide where to cut. So as type 1 I have stated, so this is the type of restriction endonuclease which has a recognition site, but its cleavage is random. It can cleave randomly at a far distance, anywhere it can cleave, far from this recognition site. But when we talk about type 2 restriction endonucleases, then they have particular recognition sequence, and in that particular sequence only they have a cleavage pattern. For example, in this case, as we can see, it cuts here. So this is a hexameric sequence which is AAGCTT. So it cleaves at A. After A it will make a cut, and then there will be a formation of a sticky end. So when we say there are 2 ends which can be produced, one is sticky end, other is blunt end. So when we have a recognition sequence which is composed of, for example, 6 nucleotides, and if cut is such that it is not equally distributed among both the ends, then that type of cut is known as sticky end. So this cut is a sticky cut, whereas if the cut is in this way, for example, we have 6 nucleotides and cut is placed after 3 nucleotides. So now there is balance among both the ends. That means 3 and 3 nucleotides are overhanging. So there is a balance. So this balance cut is known as blunt cut, or it is known as blunt end, where there is no overhang, greater overhang on one end. So where there is same number of nucleotides on both the sides. So now these type 2, they cut at this particular recognition site. They have some cleavage pattern which can vary among different types. Then comes type 3, which cleaves at a distance from the recognition site. For example, we have this EcoP15I, which is type 3 restriction endonuclease. So this is the recognition site, which is CAG. So whenever this enzyme identifies this site, it will cleave after 25 to 27 base pair downstream. So this distance is fixed. This is the only difference between the type 3 and type 1. Both type 1 and type 3 do not cut at recognition site, but for type 1, the cut site could be randomly present, whereas for type 3, the cut site is decided. It is present at some distance. For example, for this case, it is fixed after 25 to 27 base pairs. So after the types of restriction enzymes, there is also the types which is based on the recognition and the cleavage. So one such type is isosizomers, which has same recognition site and makes the same cut. So if we talk about SPH1 and BBU1, these are the isosizomers in which they have the same recognition site. So both have same recognition site and same cut is being produced. Then comes neosizomers, in which the recognition site is same, but the cut which they make is different. For example, SMA1 and XMA1. So in this case, as you can see, the recognition site is exactly identical. That means 3G and 3C. But this SMA1, it cuts after 3Gs. That means it makes a blunt end. Whereas if we talk about XMA1, then it cuts after 1G. So that means it will have the overhang towards its other end, due to which it makes the sticky ends. And then the third type is the isochordamers, in which the sequences are different but they make same cohesive ends, such as VAMH1 and MBO1. So in this, although the recognition site is different, but after the cut, the overhang, as you can see in this case also it is GATC, whereas in this case it's also GATC. So now they are having similar cohesive ends. So now what is the advantage of this cut? So after the cut is being made, what can happen is that I can also cut my DNA of interest with the same restriction enzymes, and now as these sequences, these nucleotides, so as in both of them we are making the same cut, so these terminal sequences, they will be complementary to each other, due to which they can ligate to each other. So that's why they are known as cohesive ends. That means they are helping to ligate, they are joining something. So maybe in the next slides we'll see that how the joining is happening. Right now it might not be clear that how this cutting is helping us to join the new segment into the already available vector or the plasmid sequences. So now next is DNA ligases. So now we have covered restriction enzymes which are responsible for the cut. Then we are talking about DNA ligase. So now as we have made the cut, we are joining the recombinant DNA, we are constructing the recombinant DNA, but another thing is now we also need to join them. After cutting, we also need to seal them. So for that we use DNA ligases, which are also known as the molecular glue, which join DNA fragments by phosphodiester bond. So whenever the cut is made, it is made between the sugar phosphate backbone. So the bond which is broken down is the phosphodiester bond. So they helps to join that two sugars by the phosphodiester bond. And in E. coli ligase, it uses NAD positive, whereas T4 ligases, they use ATP. So there is some energy which is required to seal these breaks. Then comes polynucleotide kinase, which is responsible to add the phosphate to 5' OH end of DNA and RNA, and these are used for end labeling as well as repair processes. So as we know that DNA runs from 5' end to 3' end, so we also need 5' phosphate group at our 5' end for labeling it, so for which we use polynucleotide kinase. Then comes alkaline phosphatase, which removes this 5' phosphate group and prevents the vector from self-ligation during the cloning process. For example, BAP, which is bacterial, CIP, which is calf, and SAP, which is shrimp. So there are different types of alkaline phosphatases with respect to each individual. They could have different names, but the main working of this this particular alkaline phosphatase is to remove the phosphate group from the 5' end. So these are some of the enzymes which are used in genetic engineering, and here I have shown that alkaline phosphatase, as this have 5' phosphate group, it is removed and now it is replaced with the OH group. Whereas if we talk about polynucleotide kinase, then as the name suggests kinase are responsible for adding the phosphate group. So now they have added the phosphate group in the 5' end on both the strands. Similar for the phosphatases. Now we will talk about the second important component, which is the vector, which will carry my desired DNA or DNA of interest. So vector, a DNA molecule used to carry the foreign genetic material into the host cell for replication or expression. And in many cases you will see that vector is also known as vehicle, which is allowing the foreign DNA to sit inside it and it will take it to the host. The essential features which are required in vector are the origin of replication. So first of all, our aim is to synthesize multiple copies of our DNA of interest. So now how it can synthesize? So it should have some origin of replication so that it can replicate. It can make multiple copies of itself. So that is one of the important feature a vector should have, due to which it can replicate independently within the host. Second thing is selectable markers. So we also need to see that whether our DNA of interest is present inside that vector or not. So for which it should have some selectable markers, due to which we can select that which one of the vector is the recombinant vector and which one is the non-recombinant vector. Such as there are some of the selectable markers which are present in the plasmid, which is well-known vector which is present in the bacteria. So they have ampicillin resistance genes, tetracycline resistance genes. They act as selectable markers. Now how they will help? So when we are inserting our DNA of interest, so we will cleave this vector with the help of restriction enzymes. Now these things, these vector, they have some sites for the restriction enzymes. For example, in the case of this particular fragment, which is responsible for tetracycline resistance, it has two cleavage sites. One is for WAMH1 and another one is for SAL1. So in this case, we can use WAMH1 restriction enzyme. We can cut here. It will make a sticky end. Now in that particular region where the cut is being made, sticky end has been generated. We will bring our DNA of interest or gene of interest. We will cleave that particular gene with BAM-H1 as well, so that the sticky ends which are produced, they are the same. So similarly, gene of interest will be inserted into this fragment. Now as this particular plasmid is a recombinant plasmid, DNA of interest is inserted into this component, so it will no longer be tetracycline resistance, because we are destroying the sequence. We are inserting some foreign DNA, due to which it will no longer be tetracycline resistant. So if we grow these particular vectors in the medium which contain tetracycline, then they will die. So this is one of the tests with which we can verify that whether the DNA is recombinant, so whether this vector is recombinant or it is non-recombinant. But the issues with this was, so the plasmids, they have these resistance regions, so with which we can, when we will be testing it, so they will eventually die, which will tell us that it is recombinant. But that will not help us. So later on, another vectors were identified which were having other selectable markers, so that we will look in the further slides. Then comes multiple cloning sites. That means it should have multiple such restriction enzyme sites where we can cleave it. So because if it has only one single site, then there is no other choice. We can only insert one gene in that particular region only. So it should have multiple cloning sites. That means at multiple regions we can insert the DNA of interest, or we can decide that where we want to make the insertion. Then comes reporter genes, which is optional, which is used to visualize or confirm the gene insertions. So there are some vectors which might have GFP signal, or they might have LAC-Z genes, with which we can grow them in certain medium and they will show us some expression. They might turn the medium into different color, or they might show green fluorescent signal, with which we can check that whether they are recombined or they are non-recombined. So this is optional. Now we will see different types of vectors. So firstly, starting with the plasmid vector, which is well studied and which was the most primitive vector which was identified. So it was obtained from E. coli, and these plasmids, they are circular in nature, and these are also known as extra chromosomal material. Means the E. coli also has its genome, but other than that it also has plasmid. So it is an extra chromosomal material which is present. It is circular in nature, and the insert size Now we also need to see that how much gene of interest we can insert into it. So let's assume we have a protein which is synthesized from a gene which is very long, more than 5 kb. Then we cannot insert into plasmid. So this insert size is also very important while we are designing the recombined, we are doing this engineering. So we also need to see that the vector which we are using, whether it can take up the gene of interest or not. So it can only take up to 5 kb, and the purpose is it's used for the routine cloning and gene manipulations. If the gene size is small and we want to make certain changes, then we use it. And some of the examples of plasmid vectors are PUC19 and PBR322. So these are well known. There are other as well. And how the naming is done? So first of all, this small p stands for plasmid. It tells you what kind of vector it is. So this is plasmid. This UC stands for from where this has been obtained. So this tells about the institutes or the scientists. So for example, in case of PBR, B stands for Bolivar and Rodriguez, who have isolated this plasmid, and 322 is just the serial number. So when different plasmids were identified, so 322 was the serial number which was assigned to this particular vector. So in this way, all the namings are done in this on the basis of this particular nomenclature. Then comes M13 vector, which is also obtained from E. coli, means which the host cell in which this vector need to be inserted is E. coli. This is a circular virus. So this M13 is a bacteriophage. It is a virus, and insert size is 1 to 4 kb, and the purpose is it is used for DNA sequencing as well as production of single strand DNA molecules. And these are some of the examples. And then comes lambda phage. This is also the bacteriophage or virus, and the host cell is again E. coli. So because we know that viruses do not replicate on its own, they are live only inside the host. So these viruses, when they are entered into E. coli, they start replicating. So this is a linear virus which can insert up to 2 to 25 kb insert, and these are generally used for the genomic libraries or high insert stability. So they are quite stable, and these are some of the names. So lambda stands for, it is coming from lambda phage, and then GT10, and then EMBL. So these are the institutes where these phage have been constructed. Then comes cosmid. So now cosmid, that means it is the combination of both the plasmid as well as the phage. So it is also inserted into the E. coli, and it is a circular plasmid. Its insert size is 35 to 45 kbs. It is used for cloning the large DNA fragments, because as if we compare it with the plasmid, then from the normal plasmid, it is approximately 9 times, it's having approximately 9 times higher insert size. And the examples are supercos1 and PWE15. There might be other examples as well. Then comes BAC, which is bacterial artificial chromosome. In this case also, the host cell in which we can insert it is E. coli. This is a circular plasmid, and in this case the range is 50 to 300 kilobase pair. So now we can insert very large genes into the BAC. So currently we use these BACs and YACs, and this is used for genomic mapping, large DNA maintenance, and these are some of the examples. Then comes YAC, which is yeast artificial chromosome. In this case, it is obtained from yeast, and it can also it can only be inserted into the yeast, which is S. cerevisiae. This is one of the species of yeast, which is Saccharomyces cerevisiae, and this is a linear chromosome. So because yeast is a eukaryotic organism, so it has a linear chromosome. Its insert size is 100 to 2000 kb, so that is very big, and it is used for cloning very large genomic fragments. So now we are not just limited to one single gene, we can clone the entire genomic fragment. So that is the advantage of these bacterial artificial as well as yeast artificial chromosomes, and these are some of the examples. And in this I have shown how they look. So for example, if we talk about the TI plasmid, which is a cloning vector, so in the later slides we will see what are cloning vectors, what are expression vectors. So TI plasmid, so it is a plasmid. It is a cloning vector which is used for cloning. That means making multiple copies out of that particular gene of interest. So it should have origin of replication. It should have certain sites where we can cleave it. So this particular TI plasmid, it is also known as tumor-inducing plasmid, and it is obtained from Agrobacterium tumefaciens. So Generally, when we have synthesized the BT plants or some other uh, crops which are resistant to pests, so in that case this particular plasmid is used because it has a virulence region. uh, so if this particular plasmid is inserted into the plants or the organisms, then they will the pests which will eat that particular leaf, they will have this particular plasmid into their gut, and then this virulence region will synthesize certain toxins which can kill the pests, so due to which we can protect our crops. So we'll go into the detail in the later slides, but for now we'll see that it has different genes which can synthesize multiple proteins: auxin, cytokinin, opine, and then this is the key characteristic of this particular plasmid. Then if we talk about phage vectors, for example M13 phage, so these are different sites. It has the lac-I region, then there is a promoter region, it has the gene fragment, it has operator, so due to which it can express. So if we insert a gene of interest into it, then it can express that gene, it can make protein out of it. It is not just cloning it, it can also expressing it. So this type of vectors which are responsible for expression of gene of interest are known as expression vectors. Then there are cosmids, which are combination of both plasmid as well as the phage vectors. So the phage vector, as it is linear, it has the cos sites, and those cos sites are also known as the cohesive sites, which can join to one another and construct a circular shaped vector. So in this way, in this case it is a plasmid, but it is also having the cos sites which are coming from phage, due to which it is referred to as cosmid. It has a polylinker, as well as it has ampicillin resistance gene, which is a selectable marker due to which we can distinguish. And then these are the examples of YAC and BAC, that means yeast artificial chromosome and bacterial artificial chromosome. They also have some other sites which are present, and we can insert the larger DNAs or larger genome fragments into these types of vectors. But as you can see, most of them are circular. And in this case also, as we said that YAC is linear, but it has some sites due to which we can circularize it. So as soon as we insert the DNA of interest, it will be circularized. And in this I have shown the difference between the shuttle vectors as well as expression vectors. So shuttle vectors, the host cell is E. coli, yeast or others, whereas if we talk about expression vectors, so in their case it depends on the system in which we are expressing it. It could be E. coli, it could be mammalian, it could be yeast. So as when we were talking in the previous slides about the livestock, that genetic engineering can also be done in the livestock as well. So we can synthesize some organisms which could be genetically modified. So in that case, we can put that particular vector into the mammalian host. This mammalian host could either be sheep, goat, kettles, or mouse. So there could be any mammalian organism. Then comes yeast. So yeast is also used as an expression vector in which the gene of interest could be expressed. And both of these vectors, they are circular in nature, circular plasmids. The insert size varies from 1 to 10 kb, 1 to 10 plus kb, whereas if we talk about expression vectors, then their insert size is typically 1 to 5 kb. This is not very big. And the purpose of shuttle vector is that it transfers between two host systems. That means from one organism we can transfer it into another organism, whereas these expression vectors, they have high level of expression in which we are not shuttling among the recombinant DNA. We are not transferring it from one organism to other. We will be keeping it in the same host organism and we are just expressing in that particular host. And the examples of shuttle vectors are these: PBR322 waste hybrids and PYES2, etc. And if we talk about expression vectors, then they are like PET series, which could be PET28A, and the numbers could vary. Then PGEX, PBAD, PCDNA3. So these are some of the examples of expression vectors. But the main takeaway message from this particular slide is that cloning vectors is used for the multiplication of gene of interest. Shuttle vectors is used for the transfer of gene of interest from one host to another host. Expression vectors are used to express the gene of interest in the higher amount. And this is another slide which also includes the difference between the cloning and expression vectors, and if you're interested you can go through the slide as we have already discussed this. Okay, so now we will talk about the applications of these genetically modified organisms. So if we talk about medical therapeutics, that how this genetic engineering is helping us. So first of all, the insulin production from E. coli. So this is the first recombinant human protein which was synthesized in 1938, and in this case what was done is that human insulin gene was taken, which is our gene of interest. It was inserted into the plasmid of E. coli, and then after that the plasmid was inserted into E. coli, and E. coli is now cloning that gene of interest, synthesizing greater amount of insulin. So this has revolutionized the diabetes treatment because now insulin could be produced in greater quantity within the E. coli. Then comes hepatitis B vaccine, which was synthesized in the yeast, and this is a recombinant vaccine using yeast to express the surface antigens, which is HBs AG. AG means antigen of the hepatitis B virus. As we know that if there is antigen present, then our body synthesize antibodies against it. So in this way, similarly, hepatitis B vaccine was also generated. So this was antigenic vaccine. Then comes monoclonal antibodies. So these were synthesized within the mammalian cell lines, and these are some of the examples. For example, Herceptin, which is a famous monoclonal antibody which is used for HER2 positive breast cancer treatment. And then comes human growth hormone, which also synthesized inside E. coli, and this is used to treat the growth hormone deficiency related problems, and it is extracted from cadavers. Then TPA, tissue plasminogen activators, which is responsible for clotting. So for blood clotting, this protein is necessary. So what we can do, we can synthesize it in mammalian cell lines, and then we can use this particular TPA activator, and we'll give it to the person who is suffering from blood clotting issues. Then comes interferons, alpha, beta and gamma. So they were also synthesized within E. coli, yeast and mammalian cells, and they are used for the treatment of hepatitis C, multiple sclerosis and many cancer types. So these are the immunogenic agents which are responsible to generate the immune response, so they can be synthesized. Then erythropoietin, which is synthesized in again mammalian cell line, which is CHO cells. It stimulates red blood cell production, and it is used to treat the anemia in chronic kidney disease. So these are few of the medical therapeutic applications of genetically modified organisms. There are multiple such applications which exist, and now one by one we'll see that what is being done. So firstly we'll talk about how the insulin is produced. So now from the human cells, as we know, the entire human genome is being sequenced. We know which gene is responsible for the synthesis of insulin protein. So we can see the chromosomes. We will see in which chromosome this gene is present. We will isolate that chromosome. We will check for the gene which is responsible for insulin production, and we will break that fragment from that entire chromosome, for which we can use restriction enzymes to make the respective cut. Now on the other hand, what we will do, we will take the plasmid, we will isolate that plasmid from the bacteria, we will make the cut in the plasmid. So when the plasmid is cut, so we will see which restriction enzyme to use. So we will use one particular restriction enzyme, and that same restriction enzyme is used to cut the human gene as well. Now we will join them, and how this joining is happening, because both of them were cleaved with the same restriction enzyme, so they will have the same sticky ends, and those sticky ends are like a single stranded DNA overhangs, and DNA has this ability that whenever it finds the another complementary DNA sequence, it will quickly get renatures. So in this way this will renature and it will synthesize a circular DNA molecule. Now this particular plasmid with the gene will be inserted into some host, and this host will be a suitable bacteria. It could be some E. coli or other bacteria in which we want to replicate it. Most probably it is the E. coli, and then now it will replicate, it will divide, and it will make multiple copies of that particular gene of interest which is present in the plasmid. So now we will further increase the reproduction of bacteria as well as the plasmids, and ultimately multiple such hosts will be produced, multiple bacteria will be produced. So this particular gene of interest is now multiplicated a lot of times. So in this way the insulin is produced. Now we can use the shuttle vectors to insert into the other organism in which we can express it, and ultimately we can obtain the insulin protein which has been synthesized with this gene of interest. So this is how insulin production has been achieved with the help of genetic engineering. Then similar processes has been opted for the recombinant HB vaccine as well, in which hepatitis B virus. So this is the DNA of hepatitis B virus. Its antigen has been obtained. HB antigen producing gene was isolated, and then it was inserted into the plasmid. And now what it will do, it will make multiple copies. So in this way, multiple such antigen producing genes are synthesized. We can multiply them and we can obtain it, and we can extract and purify this. So it will be my HB vaccine, which is hepatitis B vaccine, and it is having the HB antigen producing gene, which can further be expressed or which can be obtained after this particular round of replications which have been done in the host organism. So either way they can be obtained. So in this way hepatitis B vaccine is obtained. Now third thing is the synthesis of monoclonal antibodies. So in this case also what we do is we take the antigens against which we want to synthesize these antibodies, we insert it into we immunize the mouse. So most probably they are synthesized in the rats or mouse, in which we will inject the mouse with this particular antigens. Now the mouse spleen will synthesize different antibodies against that particular antigen. After the antibodies have been obtained, they are fused with the myeloma. So myeloma are the cells which are the cancerous in nature, which divide unlimitedly. So now both of them will be fused. Now what can happen is that these antibodies, they will divide unlimitedly, multiple copies will be produced. So ultimately we can culture them and we can get more of these copies, and then they need to be screened, their expansion is done, and the monoclonal antibodies are obtained in the end. So these all methods are the genetical engineering methods with which we can achieve our gene of interest, and we can increase the number of copies of that particular gene of interest. And then there are certain scientists and their contributions towards the insulin biotechnology. So if you're interested, you can go through this list, because I've seen that in your assignment there was some question related to these scientists. So you can go through this list, that what were the main scientists involved in the insulin biotechnology. And then the one name which is more important is David Godin, who created the first recombinant human insulin. That name is Humulin in 1978 to 1982 in Genentech, because we have already discussed that the first recombinant human insulin was Humulin. So this person's name you must keep in mind, whereas the other scientists have also contributed a lot towards this insulin production. So you can also go through them. Now if we talk about the applications of genetic engineering in agriculture, then in agriculture also there are vast applications. So first GM plant which was constructed was GM tobacco, which is the system which was used was Nicotiana tabacum, which is a tobacco plant, and this was the first genetically modified plant which was constructed in 1983. It was transformed with an antibiotic resistant gene. So now if this gene was present, so later tobacco was used to express Bt toxin, which is the Bacillus thuringiensis toxin, and a viral resistance genes, which lay the foundation for further GM crop development. So these genes were expressed in this particular plant, due to which now no other pests or other worms cannot attack this plant, because it has the toxin genes which will release the toxins. So this was the foundation work which was done, and later on the Bt Bacillus thuringiensis toxin was further also inserted into other crops to improve their yield as well as to protect them from the pests and worms. So then we are talking about the Bt crops which came after this. So B thuringiensis, which is Bacillus thuringiensis, so their genes, so this is a bacteria from which the genes have been isolated and it was inserted into the crops. So crops like Bt cotton, Bt corn, they express the cry toxin. So in the later slides we'll see that how this toxin is expressed, where it is present, but for now just keep this thing in mind, that BT crop means it has a gene which is responsible for this BT toxin, and this toxin, if it is expressed, then that crop will be resistant to pests like bollworms and corn borers. Then another such example is flavor saver tomato. So previously tomato used to ripe quickly, as now also they ripe quickly. So for that, scientists have identified that which gene is responsible. So they have observed that the polygalacturonase is the gene which is responsible for the quick ripening of this tomato. So they have genetically engineered the tomato in such a way that they have designed a molecule which is antisense oligonucleotide, which can now bind with the polygalacturonase gene, and it can suppress its expression, due to which now my tomato plant shelf life is improved. Now it will be not ripened quickly, its softening will be delayed. Then comes golden rice. So now in these examples, as you have seen, that we are just protecting the crop, but we can also improve the quality. So as in case of golden rice, the rice was genetically modified with daffodil and bacterial genes. Now what happens is it has enhanced the beta carotene levels, which is a vitamin A precursor. So we can also improve the quality of the product. We can also add some new things. So in this case, golden rice, it has the greater amount of vitamin A precursors. So in this way, the people who are suffering with vitamin A deficiency, they can take this crop and they don't have to take any other drug. So this is the advantage. This is also an example of enhancement of the crops. Then comes virus resistant papaya. So in this case also, the papaya was made resistant to the virus attacks with the help of genetic engineering. Similarly, GM soybeans, so they are made herbicide resistant gene has been inserted. They were tolerant to glyphosate and simplifying the weed control and reducing the crop damage. So these are the advantages after doing the genetic engineering. Similarly, GM maize, which is GM means genetically modified maize or corn. So it has both BT genes and the stack traits. So it is now insect resistant because it has BT genes. It is herbicide tolerance. So now what happens is that there are different weeds at the plants which compete with the main crop, and they don't let main crop to grow. But if we modify that crop, then it can tolerate the herbicides and as well as drought resistance. So that means in places where there is water scarcity, it can still grow there. So for that particular reason, what is done in the background is that different crops have been studied by the scientists which were growing in the drought conditions, and they identified that which gene is responsible, which is allowing that crop to grow. So that particular gene is isolated, and that is recombinant DNA has been synthesized by inserting that gene of interest. So now the crop which is synthesized, that will also become drought resistant because it is expressing the gene which is responsible. Similar example is of Arctic apple, which is the apple with gene silencing. So now it has suppressed the PPO gene, due to which it will prevent the browning after cutting. So generally when we cut the apple, it turns brown due to the oxidation and other processes. So with the help of this particular engineering, we are silencing this gene which is leading to the browning, so which improves the freshness and appeal. So this is kind of visual thing which is done. So visually now apple will not look that it has undergone oxidation, so it will be more appealing and it will look more fresh even after cutting it for the long time. So there is one review which has been published, in which they have talked that although this GM crops are helping us, but there are certain social and physiological factors which influence their use. Many of the countries, they have banned these genetically modified crops because they believe that what is naturally available, that is most healthy, and the genetic engineering is disturbing the natural balance. So in this review, they have talked about that what are the cultural and physiological factors which are influencing the public perception of this genetically modified foods, because these genetically modified foods, they might appear fake or unnatural or artificial. And then there is also the concern about the environmental and health risks, because we know that the balance of nature is very important. So if we make the crops which are resistant to pests and all other organisms which were previously feeding on these crops, so now it will disturb the balance, so which can in future, which might lead to some environmental or health risks, because if they are destroying the pests, so in some or other way they might hurt human as well in the later terms, because for now the quick effects are not visible, but for the long term they might impact the mammals or humans as well. So there are still some health risks about them. And then mandatory labeling and public educations are suggested as effective strategies. So it is very important, if there is some genetically modified crop which is grown, then there must be proper labels and public must be educated that what are the crops which are given to them. So these are some of the key points which are mentioned, and in this case it is showing that if there is a normal plant, then you can see how the pests are roaming around, but if it is the GM plant which is of first generation, then it is pest resistant. Then comes GM plant which is second generation. So that means after this there are other genetically modifications which are going on, so which are like herbicide tolerance, altered growth and yield. Now the crops are genetically modified in such a way that their yield can also be improved, greater number is being synthesized. And then modified product quality, for example golden rice, so we are also modifying its quality. Then abiotic stress tolerance, for example we are using the drought resistant or other genes such that it can tolerate such environments as well. So in case of these GM plants of second generation, there are multiple such modifications which have been done. So now the crops, they are very far from the normal plants. So in this way, this review tells us that how we should look at these crops, and there is different perception among people related to genetically modified crops. Some believe that this is a good innovation, whereas others believe that this might disturb the balance and this is not good. So many countries, due to which they have banned it. Now there are few advantages. So till now we have discussed the advantages in therapeutics, agriculture, and these are the combined advantages. So such as faster growth and higher yield in terms of agriculture, resistance to pest and disease, improved nutritional content as we have seen in golden rice, drought and climate tolerance, so this we have also seen, reduced use of agrochemicals. So previously we need to spray the herbicides and other chemicals, but now because the crops are genetically modified, they can fight with the weed for the nutrition, so now we don't have to spray the herbicides or the pest resistance. Then medical applications, so in which we have seen that how the insulin or HB vaccine or monoclonal antibodies have been synthesized with the help of genetic engineering. Then environmental benefits, so because we are genetically modifying the crop, so now we are not using the chemicals which we were using previously. For example, previously we were using pest resistance, herbicides, we were spraying our crops, or to increase the shelf life we were giving few injections, we are spraying the plant. So now because of genetic modification we don't need to do it. So in that way it is saying that there is an environmental balance or environmental benefit out of this genetic engineering process. Then comes industrial use. So there are some microbes which can be used to produce enzymes, biofuels, biodegradable plastics. So this is the industrial use in which we can increase the production of these enzymes or biofuels by using the microbes. So now if this genetic engineering has advantages, then it also has some limitations. So now we will look into the limitations. So first limitation is the health risks and allergies. So because if we are inserting the foreign genes, it might lead to some unforeseen allergic reactions because we don't know how it will impact us. Or it can also lead to long-term health effects which are not immediately evident. So this is one of the concern which many countries believe, that for now we are not seeing any immediate effects, but what if in the later future we see some effects. Then second thing is environmental impact. So now the gene flow from the GM crops to wild relatives, they can result in super weeds. As we know that with the evolution, the organisms have also evolved. As we can see that the mosquitoes, they have now evolved. They are now resistant to the primitive chemicals which were sprayed on them. So as we are designing the new crops which can tolerate extreme conditions, there is a possibility that these weeds, they can also take up those genes and they can become super weeds, and which cannot be controlled further. So there is still the problem of environmental impact which can take place. And in later we can also see that these insects, they might develop the resistance towards Bt toxin. So for now we are seeing most of the cases of tuberculosis, that previously we were able to manage the tuberculosis cases. So Mycobacterium tuberculosis is the bacteria which is responsible for causing TB. So previously the drugs or the antibiotics which were present, they were able to control TB, but with time, due to exploitation of these antibiotics, people are taking them frequently. So now these Mycobacterium tuberculosis, it has also evolved in such a way that it is becoming antibiotic resistant. So now this thing is very problematic. So same happens in the case of environmental impact, that with the genetic modifications, the things which we are controlling now, they might also get evolved to fight against it, and they can also become resistant. Then comes loss of biodiversity. So now because we are culturing certain crops, it might reduce the genetic diversity, which means the crops will become more vulnerable to future pests or diseases. Then comes ethical concerns. So because we are modifying DNA, we are engineering it, so somewhere we are like playing gods. We are doing it for ourselves and we are artificially designing it. So there is some ethical concern to it. It is not good, such as designer babies or altering the natural course of evolution, especially in humans. So what people are now thinking, so gene therapy is still going on, but still the designer baby concept is like having lot of ethical concerns. So scientists believe that there are certain genetic disorders which can be treated. So before the birth of the baby, we can sequence its genome and we can modify its genome. But how the exploitation is done? So instead of dealing with that particular disease, some people are saying that we want some genes, the gene which might be responsible for the greater expression of the brain, neurons, better neuronal functioning, or the beauty genes, or some characteristics that can be altered. So these things are not good, which can lead to some kind of ethical concerns. We are designing everything, that is not good. Then comes socio-economical inequality. That means if these GM seeds are patent, then it will lead to monopolies. Only some people will be able to access it and others will not be able to access, due to which if those people want to access it, then the price will increase, so which will disadvantages for the small farmers. Then comes labeling and consumer rights. So now these GM plants, GM crops, they are not very transparent, and in many cases they are not being labeled in some countries, due to which the consumer's autonomy and informed choice is being disturbed. One person might not know that he is buying the GM crops, so which is not right. Then there are technical challenges because it has high cost. Then there is complexity also, which is associated with the gene delivery systems, low transformation efficiency, and unpredictable expression levels, which can limit its practical applications. So these technical challenges also exist. It is not straightforward to just do the genetic engineering and get the desired characteristics. Then regulatory and approval barriers. So to synthesize these products, we also need some approvals from the health ministries or public consultations. So these all things take a lot of time, which delay the access to innovations, so due to which these things get delayed. So these are the limitations which are associated with the genetic engineering. So now we will practice MCQs, and it would be good if you could reply and if you could answer to these questions. So now my first question is, who coined the term genome? Option A, Frederick Griffith. Option B, Francis Crick. Option C, Wilhelm Ludwig Johansen. And option D is Hans Winkler. So if you know the answer, you can put it in the chat box. Okay, so Sachin and Tikeshwari has answered. So now let's see what is the, okay. So the right answer is option D, which is the Hans Winkler. So I have also put the explanation so that you must also know what other scientists have done. So as we know that Griffith, he was the one behind looking for the what is the transforming agent, what is the genetic material which is either gene or protein. So he was the one who has discovered the phenomenon of bacterial transformation. Then second scientist was Francis Crick. So we have seen Watson and Crick, who were responsible for the double helical structure of DNA. Then comes Wilhelm Ludwig Johansen. So he introduced the term gene, genotype and phenotype, but not the genome. So the correct answer is Hans Winkler, who coined the term genome in 1920 by combining the gene and chromosome. So as we know, genes are present in the chromosome, so he combined those two words and said genome. So now the second question is, for the 64 codons, which of the following statements are true? Option A, 61 codons code for amino acids and the remaining 3 are stop codons. Option B, 60 codons code for the amino acids and the remaining 4 are stop codons. Option C, 63 codons code for amino acids and the remaining 1 is stop codon. And option D, 62 codons code for amino acids and the remaining 2 are stop codons. So I guess many of you have answered option A, which is right, that 61 codons code for amino acid and 3 are the stop codons, which you have seen UAA, UAG and UGA. Now our next question is, histone DNA interactions are mediated by what type of forces? Option A, water mediated hydrogen bonds. Option B, ionic interactions. Option C, non-polar interactions. And option D, all of these. Okay, so Sachin has mentioned option B, and yeah, Tikeshwari is right, option D. So yes, there are ionic interactions, but along with that there are also water mediated hydrogen bonds as well as non-polar interactions which are involved. So now these water mediated hydrogen bonds, they are helping to stabilize the histone and DNA complex. So histone are the proteins and DNA is my nucleotide, polynucleotide. So it's a complex between DNA bases and histone residues by water bridges. And then ionic interactions are also there, which are the primary force. So because DNA is negatively charged due to the phosphate group, it interacts strongly with the positively charged lysine and arginine residues of histones. So because my histone is a protein, so it contains certain amino acids which are positively charged. So now positive and negative interactions are taking place. So this kind of interaction is ionic interaction, and these are the primary force. Then comes non-polar interactions. So this contribute to histones core stability and histone DNA packing through hydrophobic effects. So there are certain amino acids which are hydrophobic in nature inside the histone core. So these interactions are also taking place, which are known as the hydrophobic interactions. So in this way all the options are right. That means histone and DNA interactions are mediated by all these forces. Now next question is, the first type 2 restriction enzyme to be identified was option A, BAMH1, option B, EcoR1, option C, HIND2 and option D is TAC1. Okay, yes. So many of you have answered option C, which is right. So HIND2 was the first type 2 restriction enzyme which was discovered and characterized. It was identified in late 1960s and as the name HIND, that H and IND. So H stands for Haemophilus and IN stands for influenzae. So it was the first enzyme which was shown to cut the DNA at specific internal sites because it is the restriction endonuclease. So it laid the foundation for modern genetic engineering. And now if we talk about BAMH1, so it is the example of type 2 restriction enzyme from Bacillus aeroliquefaciens, but it was discovered later. Then comes EcoR1, which is also well-known type 2 restriction enzyme. It was discovered from Escherichia coli, due to which its name is ECO. It was discovered after HIND2. Similarly, if we talk about TAC1, so it is also the thermostable type 2 restriction enzyme, and it is obtained from Thermophilus aquaticus, which is used in molecular biology, but it was not the one which was first discovered. So that's why HIND2 is my right answer. But if we say type 2, then all of them are the type 2 restriction enzymes. Now my next question is, the DNA from which two organisms did Paul Berg use to create the first recombinant DNA? So option A is Agrobacterium tumefaciens and lambda phage virus. Option B is simian virus 40 and lambda phage virus. C option is Bacillus thuringiensis and E. coli, and option D is hepatitis B virus and E. coli. Okay, so yeah, you have mentioned it right. It's option B, which is simian virus 40, SV40 and lambda phage virus. So the explanation to this is that Paul Berg in 1972, he created the first recombinant DNA molecule. So he combined the DNA from two different sources. One is simian virus 40, which is a monkey virus which was used as a vector, and then another was lambda phage virus, which is a bacterial virus, bacteriophage, that infects E. coli. So this work laid the foundation for genetic engineering, and due to which the Paul work has also won Nobel Prize in chemistry in 1980. Okay, so next question is, tumor inducing plasmids are derived from which organism? Option A, Bacillus aeruginofaciens, Streptococcus gracius, Agrobacterium tumefaciens, and Escherichia coli. Yes. So many of you have mentioned option C, it is right. So tumor inducing plasmids have been obtained from the Agrobacterium tumefaciens. So we isolate its plasmid, it has the tDNA region. This is a TI plasmid, that is tumor inducing plasmid, so this tDNA region. And then we have a gene of interest which is obtained from the cellular DNA. We can clone it into the tDNA region. So tDNA region provide the site for the inserting the gene of interest. We can transform it, put it back into the this Agrobacterium tumefaciens, and we can clone it. We can increase the number of copies, and then we can insert it into the plant cell, which will ultimately result in the recombinant plant, which contains this particular gene of interest along with the tumor inducing plasmid. So if any pest will attack it, so tumor will be formed in the gut of that insect and he will die. Okay, so next question is, which enzyme catalyzes the formation of a phosphodiester bond at a single strand break? Option A, phosphodiesterase, option B, DNA ligase, option C, polynucleotide kinase, and option D is alkaline phosphatase. So now the thing on which you need to focus is the formation of phosphodiester bond. Okay, so multiple answers I have received. So the right answer is option B, which is the DNA ligase. So DNA ligase is generally used to construct the phosphodiester bond between the single strand break, and we have already, when we were discussing, then we have looked into these enzymes, that the DNA ligase is responsible for the formation of this phosphodiester bond, and that's why they are also known as molecular glue. So if we talk about phosphodiesterase, so whenever ase is there, it means it is an enzyme, and whenever it is an enzyme, we need to see what is the word which is present before the ase. So here it is phosphodiester, that means it is breaking the phosphodiester bond. Then comes DNA ligase, which means it is involved in the ligation, ligase. So it catalyzes the formation of phosphodiester bonds at the single strand breaks to join the DNA ends. If we talk about kinases, so kinases add the phosphate groups to the 5' end of DNA, but it is not sealing the nick. So nick means cut. So it is adding the phosphate group, but it is not joining them, it is not constructing the bond. Similarly, if we talk about phosphatases, then it is breaking the phosphate bond. So somebody has asked the single strand break, can you explain? So with single strand break, we mean that whenever the restriction endonucleases are cutting the double stranded DNA, so what they are cutting? They are cutting the sugar phosphate bond, due to which now there is no phosphate bond between the two sugars. So that particular cut is known as the single strand break, if they are breaking the single stranded DNA fragment. If the cut is on the both the strand, that is double strand break. So that is the single strand break, which is cut in the middle, in anywhere in the middle. Okay. Now let's move to the next question. So next question is, the genome of a lambda phage consists of option A, double stranded linear DNA, option B, single stranded linear DNA, option C, single stranded linear RNA, and option D is double stranded circular DNA. Okay. Yeah, so most of you were right, option A, which is double stranded linear DNA. So this is the linear DNA, but when we were talking about the cosmid, in which we said that it is the combination of both the phage as well as plasmid, so in that we talked about the cos sites or cohesive sites. So similarly, in case of lambda phage, we have these cos sites which are present. So generally this particular lambda phage, it is linear in nature, but due to these cos sites, it can also get circularized. So keep this thing in mind as well. But the genome is linear, so that's why option A is right. It has a double-stranded linear DNA genome which is about 48.5 kbs with these coarse ends which can allow it to circularize upon entering the host. So when it enters the host, then it gets circularized. And the other options were not right. Okay, now the next question is, who coined the term replication fork? Option A, John Keynes. Option B, Maurice Wilkins. Option C, Albrecht Kozel. And option D is Rosalind Franklin. Okay, so this we have not discussed so far, so let's see what is the right answer. Yeah, so you have guessed it right. So option A, which is John Keynes, so he coined the term replication fork after visualizing the structure of replicating DNA using autoradiography in E. coli, which provide the direct evidence of DNA replication intermediates. So this replication fork, because when he was observing with autoradiography, so the structure which he could see is like a fork. So if you are aware of the DNA replication process, then you might know that there are two, because DNA is double-stranded and from both the strands, both can act as templates. So both sides replication begins. So replication only starts from 5' to 3' directions. So we know that DNA has anti-parallel strands. One is running from 5' to 3' and other is running from 3' to 5'. So the strand which is running from 3' to 5' end, its complementary strand, that means its daughter strand which will get synthesized, that will run from 5' to 3'. So that direction is right. That means that strand will be synthesized continuously and that will act as my leading strand. But the other fragment of DNA which is running from the 5' and to 3', so its complementary will be 3' to 5'. But the replication cannot take place from 3' to 5', due to which the replication take place in small fragments which are known as Okazaki fragments, and that particular strand is known as lagging strand, because in that case the replication is taking place in opposite direction. So the structure, because for replication we need to denature our double-stranded DNA, for which we use helicase enzyme, which, for example, if this is my double-stranded DNA, so if I am using helicase, so it is getting denatured, so it is forming a structure like a bubble, and when the complementary strand will get synthesized, so there will be a structure of fork kind of a structure, due to which it is known as replication fork. So I hope in later sessions, when we will talk in other weeks, then we might look into detail that how the replication takes place. So at that point of time it will be clear. Okay, yeah, so the helicase is mostly acting, or the denaturation starts at the regions which is AT rich, because we have already discussed that AT is linked with the double bond, so it is easier to break those bonds. So the replication initiation is starting at the region which is AT rich. So this replication fork initial region will be AT rich, but helicase will move further. So in our case in eukaryotes the DNA is linear, so helicase will keep moving. So in that way, it's not that replication fork is only visible at AT rich region, it can move along with the helicase, but initiation takes place at AT rich region. Okay, now let's talk about other scientists as well. So Maurice Wilkins, he worked on X-ray diffraction studies of DNA, and he shared the Nobel Prize with Watson and Crick, but he did not coin the term replication fork. Then comes Albrecht Kozel. So he discovered 5 nucleic acid bases and histones. So these 5 nucleic acid bases are the pyrimidines and purines, which is adenine, guanine, cytosine, thymine and uracil. And along with that, he also discovered histones, which are the proteins which bind the DNA around itself, and it leads to compact packing of DNA into the chromosomic structures. So he did not name the replication fork. Then comes Rosalind Franklin, and she was the one who provided the critical X-ray diffraction data of DNA, and based on her data, those two scientists, they have discovered the double helical structure of DNA, the Watson and Crick. So she was the one. Now let's move to next question. So next question is, the flavor saver tomato has an increased shelf life due to suppression of which gene? Option A, ACC synthase gene. Option B, beta galactosidase gene. Option C, sucrose synthase gene, and option D is polygalactouranase gene. Okay, so many of you have mentioned option B, which is a beta galactosidase gene, but the right answer is polygalactouranase gene. So let's talk about all these genes. What are the works? So first of all, first gene is ACC synthase gene. It is involved in ethylene biosynthesis, which affects ripening, but in the case of flavor saver tomato, they did not suppress this gene. Then comes beta galactosidase gene, which is involved in the carbohydrate metabolism. So it has nothing to do with the fruit softening or fruit ripening in case of tomato. Then comes sucrose synthase gene, which plays the role in sugar metabolism, and it has no role in the softening of tomato. Then comes polygalactouranase gene. So in case of flavor saver tomato, to increase the shelf life and to delay the softening of this tomato, they have designed the antisense suppression of this particular gene. So when we say antisense suppression, so if we talk about the molecular biology terms, so each gene give rise to mRNA, which makes the protein. So in this case, polygalactouranase gene, it is responsible for the ripening, quick ripening. So what they have done is they have designed the antisense miRNA, which is the RNA which is complementary to the messenger RNA of this gene. Now what it will do is this, whenever this RNA or this oligonucleotide, it binds to the mRNA of this gene, it will suppress it. It will not allow the gene to express. So this is the molecular biology concept behind the genetic modification of this flavor saver tomato, so which slows the pectin degradation and it thus delays the softening. So the right answer is option D, polygalactouranase gene. Okay, so next question is, who coined the term gene? Option A, Gregor Mendel. Option B, Wilhelm Johansen. Option C, Watson and Crick. And option D, Alfred Hershey. So yeah, till now you all are aware that Wilhelm Johansen was responsible for this term. Yes, you all are right. Option B. Now next question is, histones are found in option A, eukaryotes, option B, prokaryotes, option C, both, and option D, plants. So any guesses for this question? Okay, now let's see. So the right answer is option A, which means it is only present in the eukaryotes, and it is the histones are the basic proteins which are found in eukaryotic cells that package and organize DNA into nucleosomes. If we talk about prokaryotes, then they do not have histones, with few exceptions in archaebacteria. Their DNA is organized differently. So when in our primitive classes, when we have looked into the difference between prokaryotes and eukaryotes, then one difference was also regarding the histone proteins, in which we have seen that prokaryotes do not have histones, whereas eukaryotes have histones. So in prokaryotes, the chromosome is like a thin thread like structure, whereas in eukaryotes, the genome or the chromosome is, so the DNA is wrapped around the histone proteins to construct the structures like nucleosomes, and these nucleosomes are also further tightly packed into the compact structures that are known as chromosomes. So both is also not right. Yeah, so histones proteins are not the HU proteins which are present in prokaryotes. So they are different. Histones are only present in eukaryotes. And if we talk about plants, then plants come under eukaryotes. So in this case, we have only one option to select. So that's why eukaryotes is the right answer. But that doesn't mean that plants don't have histones. They also have histones. Okay, now next question is, prokaryotic chromosomes are usually option A, both linear, option B, both circular, option C, prokaryotic circular and eukaryotic linear, option D is prokaryotic linear and eukaryotic circular. Yeah, so in this you have to tell that how the prokaryotic chromosome is. It is linear, circular. So if you have looked into the plasmid, which is Okay, yeah, so you're right. So the prokaryotic chromosome is circular in nature, whereas eukaryotic is linear in nature. And so that's why other options are not right. And with this, one more thing which I want to tell you is that so it is believed that during evolution there are different hypotheses and different dogmas which exist, in which it is believed that in our case there is an endosymbiont hypothesis which states that in the primitive times there was the prokaryotic cells, and one of the prokaryotic cell, it engulfed the another prokaryotic cell and it converted into the eukaryotic cell. So in this case it is believed that the eukaryotes, they have originated from the prokaryotes. So in our body we have the smaller prokaryotes in it. So for example, in a cell, in a eukaryotic cell, we have the mitochondria, and if we talk about the plants, then they have the chloroplast. So it is believed that these mitochondria and chloroplast were primitive prokaryotic organisms which were engulfed by the another prokaryote, and now it has converted to eukaryote. So due to which, if we see the mitochondrial genome or the chloroplast genome, then it is circular in nature, which is more like the prokaryotic genome. So this is one of the fact which I want to tell you. So keep this thing in mind, that in eukaryotes also, if we talk about mitochondrial or chloroplast genome, then they are circular in nature, and they come under the extracellular chromosomal part. Now next question is, the first recombinant DNA molecule was created by option A Herbert Boyer, option B Stanley Cohen, option C Paul Berg, and option D Werner Arber. Yes, so many of you have answered it right, Paul Berg, because we have already discussed. So now let's look at the other scientists as well. So first was the Herbert Boyer. He is the co-founder of Genentech and a pioneer in recombinant DNA technology. So we know that the Genentech organization was responsible for the synthesis of insulin, which is humulin. So they are pioneers in the recombinant DNA technology, but they were not the first to create the recombinant DNA molecule. Then comes Stanley Cohen. So he collaborated with the Boyer, which is Herbert Boyer, and developed a method to clone the recombinant DNA into bacteria, but after Paul Berg's initial creation. So Paul Berg was the first one. In 1972, he created the first recombinant DNA molecule by combining DNA from simian virus 40 and lambda phage, which is the landmark achievement. And then if we talk about Werner Arber, then he discovered restriction enzymes which made the recombinant DNA technology possible, because restriction enzyme are the key tools to construct the recombinant DNA. So option C is right. Okay, so now let's look at next question, which is type 2 restriction enzyme, which is HIND2, was derived from option A E. coli, option B Haemophilus influenzae, option C Salmonella, and option D is Bacillus subtilis. Yeah, so this question is quite straightforward, and you are right, option B, so which is Haemophilus influenzae, and it is quite evident from its name. So even if you don't have any idea, then you can look at its name and you can figure out. So this HIND2 was the first discovered restriction endonuclease enzyme isolated from Haemophilus influenzae, and it cuts the DNA molecule at a particular point by recognizing the specific sequence of 6 base pairs. So this is its recognition site, which is GTC, GAC. So these are the 6 nucleotides present at the restriction site, and the cut which it makes at exact center, due to which it makes the blunt ends. Okay, so now if we talk about E. coli, so from E. coli we obtain EcoR1 and not HIND2. Haemophilus influenzae is the right answer. If we talk about Salmonella, then the source of enzymes like Sal1 comes from Salmonella, and then from Bacillus subtilis we have other restriction enzymes such as BSUR1 and not HIND2. Yeah, so this is about this question. And now I have also kept a difference, because when I was going through this question, I realized that many times in this particular session they have mentioned about HIND2, but if you look in the literature, then most commonly HIND3 is discussed and not HIND2. So the difference between both of them is that both of them are coming from Haemophilus influenzae only. Both of them are type 2 restriction enzymes. Both of them has the 6 base pair recognition signal, but this particular site is different. So both of them has different recognition sites. If we talk about HIND2, then this is the restriction site, which is GTYRAC, and in this case Y could be any pyrimidine, which could be C or T or U, and in this case R stands for any purine, so it could be either A or G. So this is the cut, it is making a blunt end. Whereas if we talk about HIND3, then this is the sequence which it recognizes, which is AAGCTT, and it cuts at after the A, so that means it will produce the sticky end. This I have already mentioned, blunt end, sticky end, and the discovery. So it was discovered in 1968, and it is the first type 2 enzyme which was being discovered. HIND3 was discovered lately, but it is widely used in cloning, and application is because it has the historical significance as it was the first one, but it has limited modern use, whereas HIND3 is commonly used. So keep this thing in mind, because you might see that HIND3 is mostly cited in literature and HIND2 is not discussed anywhere. So this is the difference between both of them. They only differ in the recognition site and the type of cuts which they produce. Now next question is, type 2 restriction enzyme has option A 3 subunits, option B 2 identical subunits, option C 2 different subunits, and option D is 1 subunit. Okay, so let's see what is the right answer. So option B is right, which means 2 identical subunits. And in this I have provided you for all the 4 restriction enzyme classes. Mostly 3 are being discussed and 4th is not much discussed. So now we'll discuss about their structure. So if we talk about type 1, it is a multimer, that means multiple units or multiple chains are present, and it is a heterotrimer, that means there are 3 chains and they are different from one another. So that's why it's known as heterotrimer. If we talk about type 2 restriction enzyme, it is a homodimer, that means both the subunits, they are identical, so it has got its name homodimer. Similarly, type 3 is also a homodimer. And if we talk about the recognition site patterns, then for type 1 there are 2 sites in any orientation, whereas for type 2 it is generally small, 4 to 8 base pairs. So for example, in case of HIND2 and HIND3 we have seen there is 6 base pair recognition site pattern, and usually palindromic. So this palindromic term also you might come across, so that means it can read from both the sides and it is same when we read it from both the sides, that is known as palindrome. And for type 3 there are 2 sides, head to head orientation, and they are non-palindromic. And then there is weak specificity if we talk about type 4, that's why type 4 is not much discussed. And if we talk about cleavage site, then we have already seen that type A has the random cleavage site, it can cleave anywhere. If we talk about type 2, then its cleavage pattern is quite specific, it knows where to cut it, and it cuts within the recognition site only. It could either make the blunt end or sticky end. Whereas if we talk about type 3, then it cuts away from recognition site, but that distance is already fixed, it's known. For example, 24 to 25 base pairs from the recognition site. But type 4, its cleavage site is methylated only. If it is methylated, then only it can cleave, otherwise not. And then there are certain cofactors which are responsible for the functionality of these restriction enzymes. So type 1 require energy as well, as such as ATP, Mg2 positive and SAM, whereas type 2 only require Mg2 positive as a cofactor. So cofactors are like the agents which help the enzyme to function properly. Whereas type 3 also require energy in the form of ATP, and it requires Mg2 positive and SAM. And if we talk about type 4, then it requires both ATP and GTP. So I hope now it's clear. So if anybody will ask the composition of each of these enzymes, then you will be able to answer it. Okay, now let's move to next question. So M13 vector contains option A double-stranded filamentous DNA, option B single-stranded filamentous DNA, option C single-stranded filamentous RNA, and option D is double-stranded circular RNA. Okay, now let's see. So the right answer is option B, which means single-stranded filamentous DNA. So it's not RNA, it's having the DNA. So most of all the vectors which we have seen so far, they all were having the DNA molecule in it and not the RNA. So if we talk about M13 vector, this is how it is being shown. It has multiple cloning sites, and then there are different regions which are present. So I won't go into the detail, these things are not much needed, but the main thing is that it has certain genes which are responsible for the protein coat synthesis, other genes are for single-stranded DNA binding, and some are responsible for the replication, such that if gene of interest is inserted, then it can replicate and clone it. So this is how it is observed. So in this case, this is the schematic representation. It contains the single-stranded DNA, which is filamentous in nature, and the genome contains 9 genes which encodes 11 proteins. 5 of these proteins are coat proteins, because we know M13 is a bacteriophage, so it has the proteinaceous coat around it, and inside it DNA is present. So 5 of these proteins are responsible for this coat formation, while the remaining 6 proteins, they are used for the replication of the genome and assembly of the phage and phage extrusion. So I hope you have got some idea about M13 vector, M13 phage. Next question is, crytoxin is produced by? So option A, Bacillus thuringiensis, option B, Bacillus subtilis, option C, P. aeruginosa, and D is E. coli. Yeah, so many of you are aware of this thing. Yeah, so you're right. So crytoxin is produced by Bacillus thuringiensis, because when we were talking about the BT tobacco and BT crops, so we mentioned that this Bacillus thuringiensis, it has the gene which is responsible for this crytoxin synthesis, due to which it is used for construct the GM crops which are now resistant to pest. So now I'll tell you the detail that how it happens. So this is the structure of BT, which is Bacillus thuringiensis. It has some endospores, DNA, it has ribosomes, which are protein factories, and in the center you can see this is the BT toxin crystal, and it is surrounded by the cell wall. So now what happens is, whenever we insert this particular Bacillus thuringiensis, because it contains, its chromosome is taken and its gene which is responsible for this toxin crystal synthesis is also taken, and when the crop is engineered with this BT gene, so whenever a pest will eat that particular leaf or crop, what can happen is that this Bacillus thuringiensis, it has spores as well as delta endotoxins, which are represented by this diamond symbol. So pest will ingest these endotoxins, and as soon as it enters the gut of pest, so due to the change in the pH, because it will be acidic in nature, so it gets converted into protoxins, its coat will get dissolved, and now this crystal will break, this crytoxin gene will come out, it will convert to protoxin, and after some time it will get activated, it will interact with other proteins, it will act with proteases which will cut down this part. So protoxin means which is not yet toxic, which has some amino acid stretch which is not allowing it to get activated, but with the help of proteases, so these are the enzymes which cut the proteins, they will cut this and it will remove the part which is not converting the toxin into activated toxin. So this, after this chopping of this part, it get converted to activated toxin. Now this activated toxin, it binds to the binding receptors which is present at the coat, at the surface of this pest, and now because this toxin is binding to the surface, it will create pores in the cell wall, or you can say in the cell membrane which is surrounding the cells, it will make the pores, due to which now this pest will not be able to maintain the permeability, so anything can flow in and out, there will be no control, due to which after the pore formation, due to imbalance of due to lack of balance between the permeability, the insect will die. So this is how Bacillus thuringiensis crytoxin gene gets activated in the pest, and how it makes the crops pest resistant. So whenever it will happen, then the other pests will get the idea that we have not to eat this plant because it might kill us. So this is the idea behind the BT plants. Now next question is, BT plants are engineered to control option A weeds, Option B, lepidopteran pests. Option C, fungi, and option D is bacteria. Yeah, so as we have discussed about BT, Bacillus thuringiensis, so yeah, you all are right. So right answer is lepidopteran pests. It has nothing to do with the weeds or fungi or bacteria. Now next question is, ORI-C is the site of option A, transcription, option B, translation, option C, replication, and option D is cell division. Yeah, so in this also you are right, that is option C, which is replication. So when we were talking about the properties of vectors, that what are the features which we need in vectors, then we also discussed that it should have the independently replicating sites, which is the ORI-C. And here is an example for the plasmid, which is obtained from E. coli genome. So it has the ORI-C region, and this ORI-C site is responsible for replication because it has the tandem array of these GA, T, CT, and this particular sequences are present in the tandem arrays. Tandem means next to each other. That means adjacently they are present. And binding sites for DNA A proteins. So this DNA A protein in other organism, it is also called as helicase. So when we talk about, means this is the another name for helicase, DNA A protein. So as you can see, TTA, T, NCA, NA. So you can see it is having greater number of A and T residues. So these all the sites, they are recognized by DNA A protein, or we can say by the helicase protein, which will denature the DNA. And denaturation of DNA is responsible for the replication, because if we will not be able to denature it, how we will synthesize the daughter strands? So this is how the ORI-C site looks. Okay, now next question is, single nucleotide change in DNA is called option A, replication, option B, mutation, option C, transcription, and option D is translation. Yeah, so you're right, option B, which is mutation. And one more thing which I want to add, that mutation can be of multiple types, but if we talk about the single nucleotide change, then this type of mutation is known as point mutations. And there are different types of point mutations. Three main types are the silent mutations, missense mutations, and nonsense mutations. So when we say silent, so these words, they tell you about the visual characteristics of these mutations. So when we say silent, that means they have no effect on the protein sequence. So in this case, as you can see, here they have mentioned, after, in place of A, it was earlier coding for GTA, was earlier coding for valine, but now this A is being replaced with T, but now still it is also coding for the valine as well. So now there is, if you look at the amino acids which are encoded, they are same, although we can see that there is nucleotide change. So this type of change, in which we do not see any change at the protein level, these mutations are referred to as silent mutations. That means they are taking place, but they are silent, they are not visible at protein level. Then comes missense, which means there is change in the amino acid, so it leads to amino acid substitution. So similarly, if we talk about the third codon, which was previously CCC, forming the proline, but now this C is being replaced with A. This is the point mutation, that means C is changing to A. Now in place of proline, it is synthesizing threonine, so due to which the amino acids have changed, so the sequence is different. So this type of change is known as missense, that there is some change. Then comes nonsense. Nonsense means when a stop codon is being substituted for a amino acid. So previously it was TAC, but if this C is replaced with G, it makes TAG, which will be a stop codon. So when we were looking into the codon table, then we were looking at the level of RNA, we were looking at the U, but this TAG, if this particular DNA synthesizes the mRNA, then that particular mRNA will have UAG. In place of T, it will have U. So due to which UAG is my stop codon. So in this case, because we are looking at the level of DNA, so we are calling it as stop. So in this case, whenever this mutation takes place, so it substitutes the amino acid with the stop codon, and when we will translate amino acid, we will translate this particular mRNA, then it will lead to the premature termination of my protein. So instead of tyrosine, stop codon will come, and later on translation will not take place. So these are some types of point mutations. Okay, so next question is genes are located on option A ER, which is okay, so ER cytoplasm and chromosomes and cell wall. So you need to tell where the genes are located. Okay, so the right answer is option C, which is chromosomes. So because if we talk about the ER, which is the endoplasmic reticulum, so in that case we don't have genes because it is present outside the nucleus. Then comes cytoplasm, so this is also outside the nucleus, whereas genes are located in the chromosomes. And then comes cell wall, so not cell wall is also not the right answer, and these are the chromosomes. And this is a DNA molecule in which different genes are located. Yeah, so this is quite straightforward. Now let's look for the next question, which is question number 23. So first genetically modified plant was BT cotton, tobacco, tomato or brinjal? Yeah, so you are right, because this also we have discussed that tobacco was the first genetically modified plant and it was developed in 1983. And in this case, scientists introduced a gene for antibiotic resistance into the tobacco plant using the Agrobacterium tumefaciens, which is a natural plant genetic engineer. And due to this successful integration, it became the first plant to be genetically engineered, and after this it has laid the foundation for the construction of further BT plants such as BT cotton, GM tomato, BT brinjal. And in this I have shown the applications of these plants. So if you currently see, then BT tobacco is not discussed anywhere. So it started the construction of genetically modified plants, but now it's no longer synthesized. Then comes BT cotton, which is pest resistant, herbicide resistant and high. It also has improved the yield and it is resistant to bollworm infestations. Then flavor saver tomato, so it has increased shelf life, which means delayed ripening, and its nutrient quality has also improved. If we talk about golden rice, then this is vitamin A rich. If we talk about potato, then higher protein content. If we talk about corn and brinjal, so these transgenic plants, they are insect resistant. And if we talk about soybean and maize, they are now herbicide resistance. So these are the applications which are shown for different transgenic plants and not just the BT plants. Okay, next question is insulin production uses option A recombinant RNA, option B recombinant DNA, option C nanotechnology, and option D is bioremediation. Yeah, so you're right. Option B is the right answer, and we have already discussed that how the insulin is being produced. So it uses the recombinant DNA technology. DNA is being used. Okay, so next question is restriction exonucleases hydrolyze nucleotides in option A only 3' to 5' direction, option B only 5' to 3', option C either 5' to 3' or 3' to 5', and option D is 3' to 6'. Okay, so you're right, either 5' to 3' or 3' to 5'. So first of all, option D is completely incorrect. 6' is not even present in the DNA. So if we talk about this 5' to 3' and 3' to 5', so in the name it's mentioned exonucleases. That means it is cutting from the ends. So exonucleases can cut from either directions, either from 5' to 3' or 3' to 5'. So option C is right. Next question is restriction enzymes recognizing the same site but cutting differently are called. Option A isosizomers, option B neosizomers, option C isocodamers, and option D is neocodamers. Okay, so you both are right. Option B is the right answer, which is neosizomers. So neosizomers are the restriction enzymes which recognize the same DNA sequence but cut at different positions. So as we can see in the case of SMA1 and XMA1, both of them recognize the identical recognition sequence, but SMA1 makes the blunt end whereas XMA1 makes sticky end. And in the isosizomers, as we have discussed, they have identical recognition site and they make identical cut. If we talk about isocodamers, then we can see that although they have different recognition sites, but the cut they produce, they make same sticky ends. So in this case also GATC and GATC in isocodamers remains the same. Yeah, so the right answer was option B. Next question is, what term describes prokaryotic gene clusters transcribed together? Option A, operon, option B, exon, option C, transcriptome, and option D is intron. So yeah, you're right. So yes, so it's operon. This is the operon structure, and I guess before in our bachelor's and in our other classes also we have looked into the operon concept, that how it works. So this operon is like, we have structural genes, along with this we have operator, promoter and activator protein binding site, which is responsible for the transcription and translation of these genes. So this entire unit is referred to as operon, and it is present in the prokaryotes. Now next question is, flavor saver tomatoes were engineered to: option A, enhance color, option B, prevent softening, option C, resist pests, and option D, increase vitamins. So yes, this is also quite straightforward. It was engineered to prevent the softening. Okay. Now the next question is, which experiment confirmed that DNA carries genetic information using the bacteriophages? Option A, Griffiths, option B, Avery McCarty, option C, Hershey Chase, and option D, Michelson and Stahl. So you need to tell that out of them which scientists worked with bacteriophages and then they validated that DNA is the genetic material. Okay, so the right answer is option C and not A and B. So in case of Hershey and Chase, they worked with the bacteriophages, in which they have radioactively labeled. So once they have labeled the protein coat, and in other case they have labeled the DNA. So they observed in the end that proteins are not the genetic materials, because after performing the study they realized that no radioactivity enters the cell if we radioactively label the protein coat, but if we radioactively label the DNA, then in that case radioactivity enters the cell, which means that only the DNA is entering inside the cell and protein coat remains outside. So with the help of Hershey and Chase experiment of bacteriophages, this experiment was the last experiment, and it validated that yes, DNA is the genetic material. And now the last question for today's session is, which term refers to regions in the nucleus where rRNA is synthesized? Option A, nucleosomes, option B, nucleolus, option C, centrosome, and option D is chromocenter. Yes, you both are right. So the right answer is option B, which is nucleolus. So this bigger region is nucleus, and inside it this smaller circular region is nucleolus. So nucleolus, rRNA is generally synthesized and then it comes out, and we know that the ribosomes, they are composed of these rRNA. So this ribosomal RNA is responsible for synthesis of ribosomes, and we know that these ribosomes are further responsible for the synthesis of proteins, because these ribosomes bind to the mRNA, and then tRNA comes in, and tRNA brings the amino acids, because tRNA gets charged with amino acid, and then ultimately with respect to the codons which are present in mRNA, tRNA brings the amino acid, and these amino acid chains are getting linked inside these larger subunit of ribosome. So that is the translation machinery, how it works. So yes, B is the right answer. Yeah, so this is it for today's session. So if you have any doubts, then you can ask. Otherwise, I'll end the session. Thank you very much. Okay, so there are no doubts so far. Okay, thank you very much for answering the questions. Yeah, I'll be uploading the video of today's session on YouTube. I'll be sharing the link as well. And these PPTs will also be uploaded into the drive. And all these links will be available in the NPTEL site. So in the bottom, you will be able to find the notes and everything. And if you'll not be able to find, then do let me know. And for the next session, if you will be having any doubts for this week's assignment, then you can ask them in next week's session. So good luck for the assignments, and I'll meet you on next Tuesday. Thank you very much. and I'll be ending the session now.